From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 22nd, 2025. Hello once again, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 94th episode of Matters Microbial. As always, I am so very grateful for your listens and views and questions. Last week was graduation at the University of Puget Sound, and I was delighted to see one of my Doc Martian undergraduate students graduate with high honors. As you can see, she wore this microbial scarf by Michelle Banks to represent microbiology and is off to a master's program in
Microbe TV,
bioinformatics.
this is Matters Microbial.
Michelle makes
a podcast
wonderful
about the wonders of
microbially themed
microbiology,
art. I'll put a link to her website in the
and
show
microbial
notes.
centrism. This episode
As I record this session, I'm just back from Salem,
April
Oregon,
29th,
but I do have some microbial mascots with me at all times. It's important to represent. I was delighted to see my youngest son graduate from Willamette University summa cum laude in mathematics and physics, as seen here. He's visible in the lower right of the photograph. My son is off to earn his PhD in mathematics in a month or two, and I'm very proud. In fact, I am a Willamette dad, as you can see, even though neither of my sons studied microbiology or attended my own institution. Whoops. One of our first podcasts explored the idea of the holobiome and how every organism is made up not only of cells of that organism, but the associated microbes with it. Out of many, one. My wife, Dr. Jennifer Quinn, made me this fun mask to truly represent the Holobiont concept for Halloween. In any event, I'm delighted to welcome two guests to this podcast from Pennsylvania State University. First is Dr. Seth Bordenstein, a prior guest of this podcast, who is director of the One Health Microbiome Center, as well as Huck Chair of Microbiome Sciences and Professor of Biology and Entomology. Many hats indeed. Also visiting is Dr. Nichole Ginnan, who was first author of a very recent publication we will chat about today. Dr. Ginnan is Research Project Manager of the One Health Microbiome Center. Welcome to the Quality Quorum today, Nichole and Seth. It's so wonderful to see you.
Good to be here, Mark. Always nice to join you, my friend, and looking forward to another episode.
And I really appreciate your time. Get ready for the Seth and Mark show, Nichole. That's all I can say.
It's dangerous.
I'm excited.
Seth, you've been on the podcast before, and I'm excited to have you visit again to discuss an article that Nichole was first author of. That's poor English, but you know what I mean. Nichole, it's wonderful to meet you electronically, and you and I, we have to talk about rhizosphere sometime.
Yes, definitely.
It's a big deal to me. So here we are, and I'm excited to talk about it. I think that the first thing to do, and either of you can do this, is talk about the One Microbiome Center.
Yeah, sure. I'll take that. So we are dialing in from Penn State University in the middle of Pennsylvania, which over the last decade plus has accumulated a very large number of faculty, students, trainees, and resources around the microbiome sciences. So even before my time, because I got here 3 years ago, Penn State said, let's go big in this area. So we're currently the One Health Microbiome Center composed of 125 faculty, 550 members from students to faculty, with all of the resources and playground one could imagine they would want. And the reason we're called One Health Microbiome Center is because we have experts in the three domains of One Health: humans, ag, and environment. And we have a deep base of scholarship coming from each of those. Our aspiration then is to merge these different microbiome domains across these three ecosystems into a unified set of scientists, community, and efforts. And that's where our name really operates.
Nichole, do you think you could take a moment and talk about like the concept of One Health in a little bit of detail?
The One Health framework really focuses on the things that connect different systems to each other, so such as environment, agricultural systems, and human health systems. And from our perspective at the One Health Microbiome Center, we believe that microbes are the base of the biosphere, and they are flowing through these different systems and connecting global health, but not just through pathogenic disease and antimicrobial disease resistance, as it has been primarily focused on before, but also potentially beneficial microbes or microbes that maybe do something in one system but not the other, but they're still flowing through and connecting.
I really like the way you say this. And when I've talked about microbiomes and having to do with the intestine, we're just a tube, that old joke. It's true. And you have the organisms that have co-evolved with us. We have things that we perhaps ingested that are just passing through, sorry for the pun. And then we have others that temporarily set up shop, sorry to follow up the pun with another one, squatters I call them, that sometimes cause issues. But it's the co-evolved stuff that interests me. And on top of that is you have these new organisms coming in and how they interact with what's already there. And that's why I'm excited about what this paper of yours is. And I believe I'd like to know what motivated you to write it.
There's two motivations. One is that we at the center are really trying to unsilo and unite the microbiome sciences. So for too long, starting from the germ theory of disease to today, in some ways, we've had siloed structures in thinking. Pathogens are separate from mutualists and all the things in between. But where One Health began is it began with a disease-centric pathogen base. That is, pathogens move from, let's say, animals to humans. Mm-hmm. And as Nichole mentioned, COVID's a good example of that from the origins of bats. And HIV is an example of that from the origins of primates to humans. But, in a world where we now appreciate the great depths of microbial diversity within us and across the biosphere, it's really time we kind of graduate that germ theory-centric aspect of One Health to one that's more holistic around the microbiome sciences. That truly does unite and unsilo these categories of the types of microbes, because in effect, they're all present together in various ecosystems, from our own insides to the outsides of the soil and plants and livestock and crops that are there. So what we see is that One Health is defined by the flow of microbes between ecosystems, and they shape the risk of health and disease. And it's not just about the pathogens, it's about the commensals and the mutualists as well. And I think by just opening up that box a little bit wider for everybody to be a part of, I think we can bring our communities together and maybe create some system agnostic principles that derive from this kind of more holistic thinking. And Nichole and I and the team of authors from Penn State One Health Microbiome Center really spent a lot of time thinking about, okay, if we open up that conceptual box, what does it mean? And we went to work on describing some of the system agnostic principles that could be exemplars for future development and the future of the field.
I think for me personally, I'm really excited about this idea of unifying the microbiome sciences and doing more comparative studies across systems, since we do know that microbes are shared across all of these systems. And like Seth mentioned, we should be able to see different unifying principles across these systems, such as how microbes respond to climate change across systems or respond to disease across systems. And if we can understand those basic principles of microbial life, then that's when we get to a point where we might be able to control them or manage them in a way that could be really beneficial for us.
Friend of the podcast, Arturo Casadevall, is not a fan of the term pathogen, and the reason for that is it's context dependent. So, for example, we have E. coli in our gut that makes certain vitamins for us. If we're in a serious accident and that organism is released into the peritoneal cavity, we can have serious sepsis. So just as a real estate agent says it's all about location, 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 I would argue with that kind of interaction between members of a partnership, it's context, context, context.
Absolutely, yeah. And I think that beautifully embraces this idea that if we decontextualize microbes, we demystify them as always being a pathogen or always being a mutualist because they potentially can be both, then that continues to open up the door of a One Health type microbiome framework where we're a little bit more free to think about all possibilities, context dependencies, and the forces that shift microbes into a pathogenic state or a beneficial state or a commensal state. And in doing so, I think we bring everybody together in that process and hopefully open up more ideating and creativity around principles that define the microbial base of the biosphere rather than siloed pathogenic things away from other things.
You know, every year I make a course T-shirt for my microbiology course. And one year, indeed, that was the model, base of the biosphere. I have to find one of those for you and send it to you.
Love that. I've heard Margaret McFall the guy use that term as well, which is just great.
Well, I don't know. Perhaps that's a mutualistic association.
Yes, it could be. I know you guys have talked a lot.
Yeah. But, you know.
Yeah.
That's Margaret.
Hero.
I want to say one more thing if I could, and I think this is important for listeners and viewers. This idea of de-siloing aspects of science is important. Now, it's super important for this paper that Nichole and company wrote, but there's always a battle in every biology department worldwide between, and I hate this expression, little bio and big bio. And in fact, when I was an undergraduate at UCLA, when dinosaurs ruled the earth, I was a cell and molecular biology type person, but I took the population and evolutionary biology requirements from that track because I said to myself, I'm a biologist. And what I really appreciate about what you're doing here is you're expanding it out. And I think that's the future right there. I mean, all questions essentially come down to ecology. I hate to say it.
Yeah, that's definitely my perspective too. And I think it's really great, especially at the One Health Microbiome Center, that we have so many diverse views of the microbiome and people taking all these different approaches from different systems and perspectives, and from an evolutionary perspective, ecology perspective, more molecular focused. And with this paper in particular, we were able to bring together co-authors who represent all those diverse communities, but still have an interest in the microbiome. And that's what we're trying to build moving forward. Part of One Health is also building up really diverse teams with different perspectives and helping to unsilo those different areas.
Yeah, I'll just follow that there were 7 authors on this paper and everyone was from a different department. So 7 departments represented. And, you know, in continuing on this theme that Nichole's broadening out, we really think at the center that the best days of microbiome sciences is not behind us, but ahead of us. And of course, we had the hype and hope phase of the last 10, 15 years. But the opportunity right now is to formalize and to solidify this as a real subdiscipline of biology. And so we spend a lot of time thinking about bringing together our diverse community, because we have to. We represent a diverse set of microbiome scientists here. We can't choose a winner in a field, in a sense. And B, it's because we are in a maturing state of the field that's going to benefit from crystallizing ideas, principles, and creating convergence out of those for the textbooks and for research endeavors.
Well, look at both of you and your own research expertises. If you think about it, Seth has looked at certain symbiotic associations between an intracellular bacterium and insects, and Nichole has looked at bacteria of the rhizosphere of plants. So once again, rather than just looking at what's on a Petri dish. And I believe me, I like me some Petri dishes. But the point I'm trying to get at is that I think historically you learn more from bringing different groups together. The very field of molecular biology was at least partially founded by quantum physicists who left that field.
Yeah, right.
And they brought their perspectives into it, like Max Delbrück and similar people.
Yeah. Love that. Great analogy there. And it is important to look to other fields to kind of see how their course of development and history, led to fragments becoming converged into one. And I think the field as a whole benefits from that structure and that development over time, rather than trying to prioritize human microbiomes over ag microbiomes or pathogens over mutualists and so forth. And it's just a wonderful time to have a group of people who have a community feeling that we need to bind together to create, a more conventionalized field going forward.
It's an exciting time to be part of that. Oh, what were you going to say, Nichole? I'm sorry.
Oh, that's great. I was just going to say one thing too is I think across the field we're getting better at interdisciplinary research, which team science in itself is still an active area of research. How can we build better teams? How can we interact better and share terminology and understand each other better? And as that field develops, our teams get stronger and we get better at being more interdisciplinary too.
I think it's an important aspect of, and I use this term directly, diversity of viewpoint, because you really have to look at different things. We talk a lot about the strength of diversity. Well, here's intellectual diversity and how important that is to bring people forward.
Yeah, well said. And at a critical time too, when there's so many new ideas in the literature and so many emphasis points. It's an exploding literature. There's something like 200,000 papers now with the word microbiome in it. How do we kind of keep track of that? And there is going to be some layering and structuring ultimately into a field that starts with we appreciate everything and then divides down, rather than starts with an extreme focus on one ecosystem's microbiome versus another. And I like to think about this even from the origin of the microbiome field. We often forget that amongst the craze of the human microbiome sciences, the microbiome revolution was born in the early 2000s from marine microbiologists. In fact, I want to give Mitch Sogin's lab credit here, because his lab published the first high-throughput 16S amplicon sequencing analysis that ultimately showed us there's far more greater diversity in hydrothermal vents at the bottom of the ocean than anybody expected. And guess what? The field followed, and everybody started to find this. And it really was an example of the field was born out of one ecosystem, has matured in others, and now it's time to bring it all together.
I'm not going to go old Thomas Kuhn on you with the structure of scientific revolutions,
I love
but
Thomas Kuhn.
It is relevant. And here's something I want to really watch Nichole's face with this. Where I used to teach, and I was the only microbiologist there, and I knew a fella, a colleague, who studied crabs, marine crabs. And he would say, and I would ask, there are lots of microbes that use chitin, and that makes up the exoskeleton of crabs. And he would say, well, I know there aren't any bacteria in there. And I'm going, and I had matured a little bit since graduate school, but I was still not quite as civilized as I've tried to be. And I said, well, how would you know that? And he said, and I quote, I streaked out on LB plates.
Oh, no.
Now, the reason this is important for listeners and viewers is that there is an idea that you study microbes by just what grows in the laboratory. And that gives you just a teeny peek through the keyhole of diversity. And that's where I'm going to call it a computational or a bioinformatics revolution that allows us to look at a lot of things on all different levels. And we've had lots of episodes of this podcast about that. And it sounds like I'm making fun of my colleague, and maybe I sort of was, but the important take-home lesson was that's how he was trained. So these things have to change over time. You have to essentially educate new ideas, and sometimes as one grows older, one is less receptive to change.
Yeah, that's really interesting. And it makes sense because a lot of times people only think about the things that they can see or can culture, but we know now that the majority of microbes are unculturable. And anywhere from any environment, only between 5 to, I think, around 40% of microbes are culturable. And on top of that, you have other complexities. So in the soil, you have a lot of relic DNA, or you have microbes where they're alive and they're there, but maybe they're dormant and they're not active. And that's part of what makes microbiome sciences really exciting, is because there's all this complexity that we're trying to decipher and understand.
I would argue that perhaps, and I'm not disagreeing with you at all. I mean, I'm a convert, but fellow traveler. But what I would fellow microbial traveler, what I would say is that we're used to the tools we're familiar with, and we kind of get our heads into that. So you probably have heard me say at one time or the other the dangers of eukaryocentrism. The fact that we focus most of our teaching on eukaryotes, at least for the intro stuff. I believe that microbial science should be in every introductory course, personally, but
Right.
I've gotten no traction with that, Seth.
No, it has to be. Yeah.
Secondarily, oxycentrism. Look at the very metabolism that we focus on is based on oxygen. But third is relevant to what we're talking about right now, Nichole. I call it colicentrism, the idea that all microbes act like E. coli. I am not dissing E. coli. I'm not dissing aerobic respiration. I'm not dissing eukaryotes. I'm very fond of 3 of them. What I am trying to say is that it limits our views. Just like single colonies in pure cultures were very useful, but gave us just a tiny peek at what was going on.
Exactly. And it also, even if you can culture something and you have it in pure culture, you're now disregarding all of its functions when it's interacting with its other community members.
Which gets us back to the context dependency we were talking about a few minutes ago. Yeah. And when we think about community, we got to think about not only the microbes interacting with microbes, but the microbes interacting with hosts that house them, in effect. And transitions between microbes living in the free living world and then microbes living in a dependent world in which they may scavenge or depend on nutrients from their hosts. And viewing hosts as more complex than just one organism, but they themselves are communities, kind of brings this all around the same theme.
Do I hear a hint of a concept, Seth?
I was trying to cue you up, my friend. You know, we work pretty well together, that we should not be in the same Zoom room together.
Well, I believe the idea is can natural selection act on groups of things? And no one has a trouble with that in some forms of ecology, no. But we're more than the sum of our parts. Everything working together is what's taking place. And on the podcast, we've talked about issues of that, how the host genome talks to the microbiome and vice versa. We've talked about the fact that sometimes the microbiome that we have within our gut can impact how well particular therapeutic drugs work. It's all part of a complex series of niches. As the saying I like so much from a professor at UC Riverside, all organisms are just bags of other organisms walking around.
Yeah, and no organism lives alone.
That's right.
It really has never. They've never been alone since almost the beginning of life, right? So there's always a community of cells. There's always a community of species that we're exposed to. And what the microbiome revolution did do, it taught us a lot about the depth of those interactions, the scales of those interactions, and how much more complex they are beyond an E. coli cell and beyond binary interactions between 2 microbes. But oftentimes, we're talking about hundreds to thousands of microbes of different types and their hosts.
So I know, Nichole, that you're familiar with Joe Handelsman's Thor assemblies.
Hmm.
And those are 4 organisms, and you look at all the different phenotypes associated with those 4 organisms working together. Now, just imagine looking at, for example, what's in the human gut, right?
Yeah, exactly.
And that's why coming up with a pattern, a framework, a way to look at things becomes so important. Is that what motivated you, Nichole?
Yeah, that's a huge part of it. So that way we can identify how we can then potentially control these communities, and also just to understand how they will be impacted upon disturbance or upon um, other things that they could encounter.
I have a saying that the hand of Darwin is on all that lives. And it's really true. You know, I also call it Darwinowing because that happens as well. And that's one of the issues in my own field, my own expertise, that's been a problem. In the process of taking things from nature and growing in the lab, they change and they evolve. And it's not necessarily a surprise. And so looking at communities becomes the big ticket item. I've often said that the most complex and most interesting topics in the world, and I am neither of these things, are ecology and neuroscience. And there are commonalities between them. And that's what I think you folks are doing in this wonderful paper, which we will link to in the show notes. And I hope people will have a look and think about what's being said, because too often, my opinion, we see media hype over, I wish I had a more eloquent way of putting it, but like, here's this magic thing that you take that will cure your ills. And it's not that. It can't be that. And it isn't.
Yeah. And I think there is a reality of how difficult it is to manage microbiomes to make an environment or an animal or a plant better. I mean, this is not easy science. It requires a lot of laboratory work. And I think we are, in many respects, on the cusp of seeing the fruits of that challenging labor in the next 10 years. And that's going to be a wonderful phase of microbiome sciences when we start seeing products on the market repeatedly. We have several investigators here in the One Health Microbiome Center that are looking at improving agriculture with microbial communities, improving oral health and gut health with microbial communities. And these are all the byproduct of, 5, 10, 15 years of effort leading to now intellectual property protection and eventually commercialization that will, I think, just be a microcosm of what the whole field is going through. There is a little bit of the, maybe we've under-delivered on microbiome therapies by now because there was so much hype and hope. But I actually don't think that it's going to stay that way for far too long from what I see in my circles here, but also beyond. I think people are still very much marching towards a better future managed through microbiomes in community context.
What would you say, Nichole?
Yeah, I definitely agree. I think that as we start to do these more comparative studies across systems and understanding the more complexities of the microbiome, we're getting closer to that goal. Probably in the last 5 years, there's been a lot of work on developing synthetic communities and really simplifying and understanding those interactions, which is a really cool direction that, I'm a big fan of. But then even more recently, with the increased usage of AI, machine learning, etc., we've now been able to make more sense of these large data sets of full natural complex communities. And so I think that with that addition of understanding the complexity, but then with the synthetic communities getting down to those molecular mechanisms, and the combination of that is what is going to really breed the kind of tools that everyone has been hoping for in the microbiome realm.
I like the word breed there, breed with the sense of host and microbes together in these contexts. But Also, I think that's part of what we've tried to push in this One Health Microbiome Sciences piece at MBio is that, we can move and embrace disease control with antimicrobial agents to a wellness and resilience control framework where we're engineering better microbial communities across systems with designer communities, synthetic design communities that will prevent the next infection or enhance the next step in agricultural output. And so One Health really does open the door to this kind of thinking that we're all sharing here, that wellness and resilience is as equally as important in microbiome changes as the spread of a particular pathogen.
And it's interesting to me to hear you both talk about this because there's so much in our heads that say, here's a magic bullet. Here's this yogurt that will cure my depression.
Yeah.
And
yeah, right.
I'm not saying that those things can't be true, although I would like to see studies on that. What I will say, it has to be a community issue. This is what I've said about the future of medicine. It's going to an infectious disease anyway. The study of infectious disease is going to be ecology. And that's just one thing. But as you say, those same principles apply at the farm. They apply in the forest. They apply at the poles. They apply in the deep ocean.
Yeah. Yeah. And for me, it's fun to think about in somewhat cautious ways to think about what are we ultimately doing when we start microbiome changes in engineering to ecosystems at these diverse scales, and what kinds of legacies will changing the microbial communities for what we perceive as our own benefit. Will they have downstream consequences that may not be the intended consequences? And of course, that is going to be a long arm of research as well to make sure the regulatory stages are in place for us to develop microbiome transplant therapies for forests, ag, humans, in whatever particular niche we're interested in. Because what we put in us and just in our own bodies is going to leave a ripple effect in how we shed those microbes elsewhere. And that's a fascinating topic and also cautionary one to think about.
There's an image that's going to come up on your screen. I had a student years ago who was taking micro from me. She's now a dentist and a wonderful one. Just kind of getting a little sleepy in class. It happens. As my wife says, I go on a bit. And I told her, I said, right now, as I'm speaking, there's a Doc Martin microbiome being emitted. And I said, and when you're breathing, that microbiome and it's like, what happens here? And her eyes got really wide. And then she went right home and I guess induced her very artistic gentleman friend to make me the portrait that you're looking at now, which is of a Snoopy style character, a Charlie Brown character. No, it's not. It's Pigpen, but rather than surrounded by dust, surrounded by microbes, because that's really how we live our lives. They're part of us. And we emit them, and they were here before us, first evolved, last extinct, as the saying goes.
Yeah.
But they're part of us.
They are.
Yeah.
And they change in real time, too, which makes it fascinatingly complex and a worthy pursuit to understand, characterize, and, see where we can make potential differences in. Traditionally, we think about structures in biology being static. From an organismal perspective, a cell is relatively static as a cell, and a tissue is relatively static as a tissue. A body, relatively. Now, of course, there's gray areas on the edges, but there's nothing like incorporating the microbiome and host together to create fundamentally a relatively static and a relatively dynamic component together that makes up form and function in the context of visible life when we have these host microbiome complexes, which of course leads us to the idea that this recognition of the reality of these complexes should be defined with nomenclature and lexicon that may lead to concepts. And of course, the holobiont biology framework has really been emerging in the last decade to give us a terminology and a set of concepts for future research around assembly, functions, and evolution of these holobiont complexes.
Seth, was there not a paper that came out many years ago now trying to lay out how we should describe microbiomes in general?
Yeah, there were many, and our work was part of that, which I think you're queuing up there. But I want to give a shout out to the many that have been in the space. Kevin Thies, who's at Wayne State University, and I published a PLOS Biology article in 2015 on the principles of holobiont biology, essentially. And we listed 10 principles that we think would define the field and lead to more inquiry and research. And those were the early days of really a fledgling idea that had come through various sources of science. Coral holobiont biology was and is very much a thing in that field. But there were folks who were trying to extend the holobiont concept to everything. And when I got a whiff of that and thought about some of the work we were doing on how animals speciate over time with their microbial communities and how those microbial communities influence directly the speciation process of these animals, in our case, they're insects, it was sort of unavoidable to think that we need a bigger appreciation of processes that diversify these structures and how elements within those structures, microbes and hosts, can influence the diversification of those structures. So yeah, since that paper came out in 2015, it generated a ton of discussion, a ton of debate. But I think what I've seen now over the last decade, because we're coming up on the 10-year anniversary, is essentially a field that has wrangled through some of these challenges, and both challenging the way people thought about siloing ecology away from evolution, about thinking about binary symbioses as the way of the world rather than these complex, loose host microbiome assemblages. I think we've kind of all grown up with something that is becoming a reality for the future of biology. So look forward to kind of talking more about that today with you.
Nichole.
Oh, thank you. I would love to bring it back a little bit to the comment you made and the image of pig pen but microbes. And one thing that we highlight in the One Health paper, and something that I think is important to highlight, is that once you start to understand that the microbes are around us and connecting us, so connecting animals to humans, to plants, to the environment, that you can start to sort of philosophically view how we're all connected, and that could lead to new avenues to promoting conservation, because healthy environments means healthy microbes that are going to create healthy spaces for humans, etc. So that it's larger than our single selves, because we are a being of many, and we share our many with many others. So I think that's something I want to highlight too.
I used to make these really cute buttons from my class where there's a face and the face is made up of microbes. And it says that I'm a microbe-biologist.
Oh.
And
Nice.
I'll see if I can't send you one of those.
I think I've got one of those in my office somewhere. Of course, I got a lot.
But it's really true, and it's philosophically it's interesting as well. And I think that I often say to students, and Nichole, I'm very interested in what you think about it. The names that we give things really impact how we think about them. And I've been struck by that in my conversations with students. Did you find that to be true as you were putting this paper together?
Yes, definitely. And that also brings us back to holobiont biology with what Seth was talking about. It can change our whole perspective, and even the word holo it makes you think of all of it instead of saying, a single organism, or a host that is colonized by microbes. You're still thinking of them separately. But when you say something like a holobiont, you begin to think about everything together, and it can change your perspective and approach to researching organisms like that.
Definitely. I like that a lot, Nichole. And the other aspect is, a holobiont structure recognizes the influence of symbiosis on life. Of course, many symbiosis and microbiologists know that already. So a knee-jerk reaction is like, yeah, what's the big deal? But the big deal is that this term isn't for any one particular discipline, right? It's for all of biology. And so when the person who doesn't think about symbiosis hears and recognizes holobiont, a cell biologist, a biochemist, a marine biologist, well, that's where Nichole's portrayal of thinking changing with terms really does matter, because we want to essentially encourage them to think symbiotically as much as we do, and vice versa. And so that term does have a ripple effect, especially outside of the core community of symbiosis biologists that recognize the importance of symbiosis.
Nichole, I don't know if you know this. Poor Seth has heard this from me several times, that there are 3 stages of scientific research. Stage 1 is, that's a silly idea. You never should have asked it. Second is, well, it might work a little bit in the lab, but it's not relevant to the natural world. And the third level was, and is, I said it was a good idea all along. And a student of mine came up with the fourth one that you'll laugh at. And I came up with the idea first. And I've seen this over and over again.
Yep. Yeah.
Love it. I love the fourth one the best. Yeah.
Well, it's funny but sad, right? Because I've seen it. Yeah. And so have you, Seth. But what I'm trying to get at here is this is a different way to kind of present this. And this is, for what it's worth, I am going to insist that my students in the fall read your paper, Nichole.
Oh, thank you.
Awesome.
Well, yeah, because it's a different way to look at it. And even in a small liberal arts institution like my own, students come here with very unusual ideas. You ask them what their favorite microbe is, and it's like they have trouble, or they will only name the one they worked with in the lab, which makes me laugh, but is true. But these things aren't in isolation. That's the thing.
So true. And we do have to, I want to come back to this theme of unite and unsilo the compartments of our microbiome sciences. Like, we have that big of a job as we do to spread the microbiome sciences outside of our community. And that's where I hope a lot of young scholarship and energy is devoted to in the next 10, 15 years, because it's needed and because it's impactful, and because we can be a better community and a vital subdiscipline of biology by embracing that.
And reflecting what it is you study.
Yeah, no doubt. And the common principles across what we all, love is our first laboratory love, for example, but these principles extend beyond.
So, Nichole, can I ask you a question?
Sure.
When you were writing this paper, what's the one thing that amazed you the most?
Good question.
Yeah.
It's a more fair question to give you ahead of time, so I apologize.
Yeah, I think there's kind of a couple things. So firstly, when I was working with all these different authors, our first draft, some of the language that we used, other people were like, oh, that's not how I would say that. Or like the terminology between the different fields, and how, even though we're all biologists at the core, that very different language is happening. And so trying to find the common language, the bridge, or making sure we're defining things that we didn't think we needed to define, that was surprising to me, because I thought we all research microbiomes. We're all in the same field, but really there's still a lot of differences, even in these closely related subdisciplines. And then the second thing is that there already are a lot of examples of microbes that are beneficial in multiple systems and travel among multiple systems. So for one example, there is a bacteria, streptomyces, that can colonize bees and produces a secondary metabolite that can inhibit fungal pathogens in beehives. And bees, when they visit flowers, they can deposit that streptomyces and it can travel down to the roots of plants and also prevent fungal diseases of plants. And seeing as I reviewed the literature that there's these multiple examples that it's already happening, and thinking about how we really do need to understand what is traveling between these systems and the impact it's having. Yeah.
A couple of things, Nichole. I collect pins, and I had one made that I'm looking at now. It says jargon excludes others.
Yeah.
And that sounds very judgmental, because it is, but it's true that you do need a particular vocabulary for a field. But as you discovered, it's possible to use language that doesn't connect more. What we need is an Esperanto for biology, and there is no such language. But we need to find a common language, and the only way that's going to happen is by starting young and getting young people thinking about this. I mean, even back to middle school. Because some of the stories that we tell, some of the wonderful tales that we tell, geosmin attracting springtails, that kind of thing, really, really interesting ideas. Leafcutter ants having a specialized structure to carry their particular Nocardia bacterium on them. All of this is fascinating stuff to get people to understand at an early age that it's a microbial world and we're part of it.
Yeah. Definitely.
Seth, you have more things to say, I'm quite sure, about this lovely topic.
Well, I do. And I think part of the paper that I really like at the end is that we reflected on the way science is funded. And even science funding is structured in a way that they silo each other away from each other. The NIH has human, the NSF has non-human for the most part, and the USDA has ag. And yet, when we scream, sort of bring us together, One Health Microbiome Sciences, we're actually going to need to rethink and redo on either interagency collaboration or on, others stepping up to provide funds that'll actually do large scale One Health Microbiome work at a level that I fear not each individual main institution today would actually be finding it sustainable or attractive, because they'll love one component of it, but they might critique the others given the nature and the history of those agencies.
I have to be a little taken aback, because in the current climate, with all of the current controversies we see about limitations on funding and questioning, it becomes so important to talk about our science in a way that's accessible to other people.
Yeah.
And I'm also very concerned about the infrastructural needs to do some of the analysis that's necessary. You're fortunate because you have a large research center. I mean, your faculty, more faculty than are at my institution.
Wow.
Humbling fact of the day. Yes.
Well, I mean, and just like when I was in school, I didn't have to learn any real bioinformatics. And now I would argue almost everyone needs to learn bioinformatics.
Yeah, right. Mm-hmm.
And so these are the new tools that we need to carry out this. I want to call it a central focus of all biology, because I would argue it is.
Mm-hmm.
And it sounds like, oh, you're just a microbial centrist, which I am. But that doesn't mean I'm wrong.
Oh, I love the way you make me smile. Yes. And it's true, right? I think you can make the argument that because microbes are the base of the biosphere, because the way Nichole described microbes flowing through these ecosystems, that they really do bind life together in a capacity that's central, that's foundational. And, whether it's One Health Microbiome Sciences, whether it's holobiont biology, whether it's education and accreditation, whether it's funding structures, these things do need to come together for all of biology, but especially the flow of microbes and microbial sciences.
Yeah, and
You know,
I would
it's
just add, I mean, I know you know this, but on my soapbox,
please.
all life came from microbes, and all multicellular organisms advanced in a world teeming with microbes and evolved around microbes to interact with them. And microbes are the most abundant life form on Earth. And so it just makes sense. Yeah,
They're the most diverse. They can do things other organisms can't. They were here first and they'll be here when we're gone.
exactly. Yes.
I mean, what I find is I tend to proselytize about this. And sometimes I need to find other ways to do that. And this is why I'm hopeful about papers like yours, because people just kind of shut that off when I go into it. I was once told by a plant biologist that I wasn't interested in anything but microbes. And I said, no, plants are plenty interesting. It's just the most fascinating things about them they stole from microbes, because they did.
Yeah, and I think you've studied it all. You know, you've studied animals and plants in your lab,
Yeah.
and you know, I do think that you have an important role to play in your advocacy, because it moves the needle away from maybe where the gravity and convention is to the other side. And that's going to force a discussion. And that discussion is what you're trying to generate so proudly and bravely, in some sense, for the folks that might doubt it, right? So I'd say thank you to Mark for your consistent advocacy on behalf of the microbes. I think you are moving the needle for microbiologists and non-microbiologists bit by bit, you know, every time you're speaking. Keep it up.
You know, you'll laugh. I hope you'll both laugh at this. A few years ago, I usually teach microbiology in the fall, and in the spring, some of my former microbiology students were taking an animal physiology course, and they were working with earthworms, and they needed to have an independent project. And they came and told me the story I'm telling you of how they were up at late at night and they were going over their independent project plans and how it would work and what happened there. And then one of them said to the other, "We haven't considered the microbial component." And then they looked at each other and said, "We drank the Kool-Aid."
Yes.
Which is why, by the way, I have microbial Kool-Aid right here.
Nice.
Oh.
Well played there, sir. Of course.
Yeah, I timed that well.
Yes.
But it is important. And so, Nichole, we had a guest on recently who was talking about how everyone should learn some degree of programming. And I mentioned that bioinformatics would be kind of important. So here's my question. As we're trying to expand how we process data, other than what we know about genome sequence and analysis, can you predict any uses of AI that we should be on the lookout for involving this?
Sure. I think that for the future, but also many people are starting to do this now, is beyond trying to just find patterns in the numbers of and the sequencing data. It's really one of the best uses for AI is to process images. So to do high throughput phenotyping at whether that's microscopic images or that's host phenotyping, such as cross sections of root to look at the anatomy, or different human cells, gut epithelial cells, and the ability for AI to then detect specific structures and measure them rather than having some poor grad student doing that with like image J. That ability to generate data in that manner is really going to impact the field a lot, and it already is starting to. Yeah, I think that's probably one of the most important applications. Another one too is that with these language models, we can input text, right? And then you can ask AI questions. And at some point, we might even be able to have a language model that can tell us what questions we haven't asked yet. Or connecting these few papers, if you know this knowledge, what questions would you have? And maybe that's things that humans haven't thought about yet. So there's a whole avenue, and I'm not an expert in AI, but I'm still very excited to continue down that path.
I'm not an expert either, but I love science fiction. And part of me kind of ran cold. I already know that ChatGPT will make up stuff from time to time, but at least we're trying out ideas. So it doesn't matter if the idea might be unusual.
Yeah.
And I need to tell you about a science fiction story from the 1940s called A Logic Named Joe, and I'll write to you about it.
Okay.
It's very relevant to what you're talking about here. That's
Great.
amazing. Thank you. Seth, a little while ago, you were discussing the idea of commercializing communities, following some of the principles you've been discussing. Perhaps the idea of trying to find communities that improve health in one situation or another, fight disease in another fashion, that kind of thing. Because you're dealing with such complexity, how could you prevent unintended consequences of introducing large numbers of things? It sounds to me, I already mentioned when I was talking a little bit to Nichole, if you look at Joe Handelsman's Thor system, you have like 3 or 4 members and this plethora of phenotypes that can happen.
Yeah, this is a wonderful question. And it's also very difficult to give a good answer to, because at the end of the day, we're talking about ecology, and ecology can have a mind of its own and a course of its own. But let's start with the idea that in simple systems, microbial changes to a particular ecosystem or organism can be done with confidence. So I'll give you an example. I study an obligate within cell bacteria called Wolbachia, and it lives in half of the world's arthropod species. And arthropods are mainly insects plus other things. And one of those insects that doesn't have Wolbachia happens to be the world's most dangerous animal on the planet. That is the Aedes aegypti mosquito that transmits Zika virus, transmits dengue virus, chikungunya virus. And it turns out Wolbachia has this mysterious ability, once it's in that mosquito by human injection into the mosquito, will block the replication of these pathogenic viruses of humans. That bacteria is then vertically inherited from mother to offspring every generation and essentially stays in that mosquito population in lineage over time. That's a well-contained microbial transplant, if you will. Now, if we're talking about engineering our gut microbial community, well, what comes in our gut also goes outside of our gut. And the problem with that is there's actually this risk of dissemination of microbes into other environments once we start modifying communities that are more open. And there will necessarily be regulatory interventions or criteria for these kinds of communities to be altered. And we're in the very early days of that. My hope is that we will see research paradigms that adjust to the necessity of looking at these microbial transplants over time and over generations, that funders will take risks to do long-term microbial transplant therapies to evaluate these risks. Seems like a necessity, right? The other aspect is to have an open view on this, that human medicine has always taken shots first to get rid of diseases and then deals with the consequences after. And of course, antibiotics is a great analog to this. If knowing that we would have ushered in the great antibiotic resistance problem, would we have stopped using antibiotics? And the answer is no. Of course, we would have used antibiotics. And so humans are very much going to do this again and again and again. We're going to take the first shots to cure the disease and then deal with the ramifications later with some amount of regulation in the process as we become more informed. I think that's the best way to look at the options, the range of modifications, and then the long-minded view on how we've always had unintended consequences in medicine over and over and over again.
I think that's well stated. And I do want to say, Seth, that when I've brought up to students the idea of introducing Wolbachia into some of the dengue-infested areas in northern Australia, and I explained what's going on,
Yeah.
many of the students had a knee-jerk reaction that this was dangerous.
Correct. Yeah.
And it's one of those things, you can't prove something is perfectly safe. So
Yeah.
this has to become an issue of relative risk.
Yeah. And indeed, the scientists behind that effort, who are some of my close colleagues, went through the process of test this in the lab first, test it in field cages next, test it in small release sites next, monitor spillover events. And so the science is there. And we need to create the space for that to be communicated effectively to the public, who often thinks about engineering life having more risk than benefit. And that's an acceptable place to start with. And it's up to scientists and the public to come together and have a good public outreach campaign around these kinds of things.
That is the biggest challenge as far as I can see it. I remember Asilomar, and it was really an issue because you can't prove a negative very well. You can't prove something is safe.
Right.
You can only generally recognized as safe. And there are many people, and I am not being judgmental of them, that have a very visceral reaction to this. And so it's our job, I think, to present things as calmly and completely as possible. I believe folks can understand much more than they believe they can.
Totally. And in fact, the Wolbachia releases were only done with a very high acceptance rate of the communities in which these Wolbachia mosquitoes were released.
That's
so they
right.
went around to doors and said, "Are you okay with this? Are you okay with this?"
Mm-hmm.
And probably got 95% acceptance rates pretty regularly, because guess what? Dengue virus is pretty bad and people want a solution. And as long as they're informed and know what's coming, I think they understand that everything in science new has a risk, but they also understand the benefit of getting rid of these devastating diseases.
Absolutely so. And that's something that I actually told the students. I said, you might want to talk to some people who've been through dengue. I said, I'm not disagreeing with you. I'm saying that it's easy at remove to say that this is a risk you would not take.
100%.
Yeah. What I want to say now, a couple of things, because I'm watching the time carefully with you both. And the first thing is, I'd like to hear, like, from both of you, your final plea, because I know that in the actual show notes, I'm going to have lots of papers for people to look at, and I'm going to tout them in the show notes as well. So what would be your final comment, Nichole, about what would be most important about this paradigm shift and why you think people, perhaps even folks who haven't thought about microbiology, why they should care so much?
Well, the world will keep changing and the climate will keep changing, and both politically and environmentally. But microbes are going to remain, and they are here. They are dispersing across systems. They are connecting health across systems, and they're essential to all life. I guess that's my main points.
I think that's a good one. They're echoes of what took place before and what's going to be happening in a way. So they can be sentinels and memories, which is an interesting idea.
Ooh,
yeah.
Nice.
Seth.
Yeah. Great question to wrap up on. I would like to answer that by reflecting on the arc of biology's history.
Please.
And I want to start with the idea that there's these three epochs or eras of biology that we have fundamentally experienced in the history and the arc of biological sciences. The first, let's start with Darwin and Wallace. And of course, we get the great evolutionary theory of life from them. And that is completely eukaryocentric and built off of their observations of animal and plant life, which, as we know, represent 1% of all life forms on the planet. And a lot of credit should go to Wallace and Darwin for the fact that what they observed in animals and plants scales into the microbial world, of which they probably knew about because there was some early knowledge about microbes then, but they obviously didn't find them important enough to write about. So The Origin of Species by Darwin was a sterile book, as we like to say. No microbes in there.
Oh, I love that.
And the second epoch or era is what I would call nucleocentrism, which is in the 1920s and 30s when Mendel's laws of genetics were rediscovered and then merged with Darwinian and Wallace theory on evolution. We get what's known as the modern synthesis of biology, where essentially biologists are explaining how organisms evolve over time through the lens of genetics, how traits vary between organisms, and how we can map that or understand that through genetic changes. And that period arguably lasted for almost a century, Mark. And the new century we're in, which is, I think, a century of holism, so we have eukaryocentrism, nucleocentrism, and holism, recognizes that both reductionist and holistic techniques around the centrality of microbes in the biological world define sort of the next 100 years of breakthroughs and radical disruption, if you will, in some ways, to what we think and know about biology now. And that, to me, is the arc of a main message for where we are in the sciences. And we spoke a little bit about this in some of our holobiology pieces, one of which is a 2024 paper in Science that we can share with you, called The Disciplinary Matrix of Holobiont Biology. And it really does capture some of this paradigm stages. And in fact, the word disciplinary matrix comes directly from Thomas Kuhn, who defined a disciplinary matrix as a social enterprise, a language enterprise, more than a data enterprise. It's when new enveloping representations really change our perceptions of how we characterize and term and conceptualize. And that's a social enterprise as much as it is a data enterprise. And that's where we are right now in the first 15, 20 years of this much more microbial oriented, but open-minded era.
You know, there's a saying from physics that physics advances on gravestones.
Oh.
And I don't like it very much. The idea being that you have to get kind of the ossified brains out of the way. And so I have found that most of the microbial scientists that I know as they grow older continue to be interested and excited by new findings. And I would encourage people to have an open mind about new ideas. I think that the big advantage to science is that you can come up with testable hypotheses.
Yeah.
Right. And especially, go ahead.
I was just going to say, to capture this open to new ideas, I mean, we are the intellectual free enterprise, right? We have to be supportive of the entrance of new ideas and their evaluation. That's totally fair too. But we need to create space for new ideas to flourish. And we need to create space for the community will define the utility of the ideas, not any one particular elite scientist or influential members in the community.
I think many people have a misunderstanding of the process of science, and that sometimes science is fault because of the way that they describe their own work. I think it's vitally important for us to be able to explain what it is and why it's important to do. There are so many examples that I used to talk to my late parents about this all the time because they would say, why is it so important to do thing X? And without even looking anything up, I could give them lots of examples. But I never learned that in a class. No one ever put that in an introductory course for me. No one ever wrote that in an essay that I found anyway.
Right.
And especially in the current climate, we need to support one another much as the holobiont that is each of us is supporting the entirety of the superorganism.
Yeah, a visible life for sure. Yeah, exactly. Well, that's a great way to wrap things up right there. Thank you, Mark.
I am very, very grateful to both of you for being on this podcast. I get excited about it because I think I talk about the one true microbial faith, and this happens to be one of them. And one, like Seth's new picture, right, is really important. And I want to wish you both every good thing.
You too.
And I hope to come out and visit and see this wonderful place someday.
Oh, you're on our radar. Don't, be careful what you ask for.
Nichole, you should be so proud of being first author on this paper. It's so impressive. and it's so clear, and more to the point, it's accessible.
I'm so happy to hear that. Thank you.
Because I look at it as an educator would in what I could get my students to read without using ChatGPT. All right?
Yeah.
Uh-huh.
So I want to thank you, and I want to wish both of you the very, very best.
All right.
Thank you.
Great to be with you always, my friend, and looking forward to the next episode, whenever it may be.
Yes.
Thank you. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as usual, truly and terrifically epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Seth Bordenstein and Dr. Nichole Ginnan can be found in the One Health Microbiome Center, Huck Institute of the Life Sciences, Pennsylvania State University, University Park, Pennsylvania. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.